and it's a great honor and pleasure for me to introduce a longtime friend and advisor to the PDB, David Searls. David received undergraduate degrees in both philosophy and the life sciences from MIT and a PhD in biology from Johns Hopkins University. During postdoctoral fellowship at the Wistar Institute in Philadelphia, he turned his attention to computational biology and obtained a master's in computer and information science from UPenn. He then spent 7 years in artificial intelligence research and development at the Unisys Corporation, including work on computational linguistic approaches to DNA sequence analysis. In 1991, he returned to Penn as a research associate professor of genetics with a secondary appointment in computer and information science, where his research centered on formal language theory applied to biological macromolecules. During this time, he founded the Intelligent Systems for Molecular Biology ISMB meeting series and was a founding board member of the International Society for Computational Biology. In 1995, he went to the dark side and assumed responsibility for leading bioinformatics at SmithKline Beecham, and which later became GlaxoSmithKline. He retired from GSK in 2008 as Senior Vice President of Computational Biology. Since then, his primary interest has been in the intersection of computer science, biology, and the humanities. So please join me in welcoming David to deliver a provocatively titled seminar, Genome as Literature. David, it's terrific to have you here, and I regret once again that we're not able to do this in person.
Well, thank you very much, Stephen. It's a pleasure to be here. Now, I'm sharing my screen. Is that coming through?
Yep.
It is, and if you go into presentation mode, you should be ready.
Okay. And now, are you seeing my...
We're seeing your retro. Please
Yes.
stand by. Absolutely. Thank you.
Okay, very good. Then I'm on my way. Well, just to be even more provocative, this is the subtitle of my talk. I need to do a little adjustment here because my screen is being blocked by the... There we go. Yeah, the title of my talk is Genome as Literature, but I've added a subtitle here that I think will spice things up a bit. The idea I'd like to explore is that there may be ways to take literally the metaphor of the genome as the book of life, which is being promoted by Francis Collins, among others. I'll be talking about my early efforts looking at how linguistics might apply to genes and macromolecules. I'm giving just a taste of a variety of lines of work, and I apologize in advance for speeding over some topics with barely a glance for the sake of providing a good overview. Then at the end, I'd like to touch on some of the more literary angles. And as a motivating example, I'm going to look at the work of Charles Lutwidge Dodgson, aka Lewis Carroll, who you know as the author of the Alice books, but in his day job, he was a mathematician at Oxford. He was also well known as a game designer and puzzle master. I'm going to take as my text his best known poem, shown here as Alice first encountered it, which required her to hold it up to a mirror. This is of course, the first stanza of Jabberwocky, on which I hope to do what literary critics call a close reading, to try to convince you that the poem is actually about molecular genetics. So let's begin by just noting that the poem is utterly nonsensical. And in fact, Carroll wrote a lot of nonsense verse. This was all the rage at the time, perhaps in reaction to the strictures of the Victorian age. Here's a stanza from another one that he wrote, and you can see it's perfectly good English, but when we try to understand it, it only boggles the mind. So Jabberwocky, in fact, serves to show that nonsense can arise at several different levels. The poem's full of completely made up words like brillig and slithy, which are just gibberish at the outset. Later, the Dada movement tried to use nonsense to promote cultural anarchy, and they promoted stringing actual words together, but in random order, which of course would seldom result in valid grammatical sentences. And as noted, Carroll's nonsense is perfectly grammatical. In fact, it's rather elegant English, but even when we know all the individual words, we can't get to grips with the meaning of the whole in any real-world sense. So this distinction between syntax and semantics in particular proves to be useful in many contexts. In fact, we see it in important organizing paradigms in many fields. The usual progression of analysis in linguistics, in fact, in computer science as well, is from the lexical to the syntactic to the semantic. And in biology, we can think of primary sequence as the lexical level, while syntax describes structures, for instance, of genes or macromolecules. And semantics refers to their function in the cell or organism, in essence, their meaning. So linguistic analysis of an English sentence entails determining its syntactic structure, what's called parsing. Starting at the bottom, we first assign categories or parts of speech to each of the words. Determiner, by the way, is just linguist talk for what we'd call an article. And then it's simply a matter of building the tree up that groups these into phrasal structures, phrasal categories at successively higher levels, ending at the notion of a valid English sentence. For reasons that'll become apparent, the words are called terminals, and the higher level categories in green here are called non-terminals. So grammar then is nothing more than the set of rewrite rules that generate such a tree, capturing both structure and relationships in a nice modular way. So a sentence comprises a noun phrase followed by a verb phrase, each of which can be rewritten at successively lower levels down to the actual ordered set of words in the sentence. And the vertical bars here mean or, that is alternative rules for each constituent. So a noun phrase is either a determiner followed by a noun or another noun phrase followed by a prepositional phrase. And finally, we bottom out at the level of parts of speech, which of course can specify a large variety of terminals or words. And this is very simple. In actual natural language grammar is going to be hugely more complex than this, but even this trivial one handles a sizable subset of English. So such a system for language generation has some properties that are important to highlight. One of these is that a parse tree captures dependencies between constituents, such as the subject, noun, and the verb, which may be separated by other clauses in the sentence. So for example, if we make the subject plural, the parse tree directs us to the verb whose number must match, and thus we have action at a distance. Another important property of grammars is recursion, the fact that a noun phrase may contain a prepositional phrase, which in turn contains another noun phrase, and so on, means that a grammar can embed any number of such phrases. And in this way, infinite languages can be defined by finite grammars. We get incredible variety from concise specifications. And finally, we have ambiguity, the fact that sentences can be interpreted in more than one way syntactically, as for example, in the case of the linguist sees the biologist with the telescope. The question is, who has the telescope? So in this parse, the prepositional phrase attaches to the object noun phrase, and this syntactic analysis of the sentence indicates that it's the linguist is seeing some biologist who has a microscope. But the same grammar can generate a very different parse tree. And in this case, the prepositional phrase attaches to the verb, indicating that the linguist has the telescope rather than the biologist. And these two alternative parses may seem like more or less of a toss-up, but if I substitute the word microscope at the end here, it tilts the balance in favor of the previous parse tree, unless we want to imagine an extremely small biologist. On the other hand, if I now substitute in the word amoeba for the biologist, it shifts the balance decidedly toward this latter parse, since it's an even bigger stretch to imagine an amoeba with a microscope. Now, in this way, semantic context tends to resolve syntactic ambiguity. Now, genes are also said to have structure, of course, and gene structure shares many of these key features of syntactic structures in language. For one thing, genes exhibit recursion, in their alternating intron-exon structure, the extent of which is only limited by practical constraints, as in language. But we also see ambiguity in gene structure, notably in the alternative splicing seen in most genes. And such alternative splicing can take many forms, even in the same gene, and this highlights the presence of dependency in gene structure, since a proper reading frame has to be maintained in all of these isoforms, and that creates constraints at a distance. So all this suggests that we should be able to write a grammar for gene structure, and indeed that's something I began doing some 30 odd years ago. And I'm showing here a toy grammar capturing only the highest level outline of recursive intron-exon structure, the details of which needn't detain us. The best way to demonstrate it is by showing parse trees, which I'll do now. One can take that grammar and plug it into a parser together with an input string and get back parse structures that even bear a vague resemblance to the usual depiction of intron-exon structures. Now, what's called a chart parser can be used to produce a table showing all possible parses should the grammar be ambiguous. So we also get back the case where the exon is skipped, as well as cases where an intron is retained, and all this from the same grammar. In other words, the grammar is compact in the same way alternately spliced genes are with respect to their transcriptional repertoire. Some more practical gene grammars that I wrote back in the day could be used as gene finders, working from first principles of gene structure. So here, yellowed with age is a sample output from parser that I wrote. For a brief while in that pre-genome era, grammar-based gene finding was actually competitive in terms of accuracy, if not speed, because exploring all the combinatorial possibilities of structures is inherently very costly, and the only reason these parsers were practical at all is that chart parsing uses a technique called memoization to save and refer back to intermediate results, and that's basically what dynamic programming does in sequence alignment. So while speed may not be a selling point of parsing as compared to purpose-built algorithms, the trade-off is versatility. Exactly the same parser could be used for a variety of other grammars, so that, for instance, one didn't have to build a separate tRNA gene finding algorithm, but simply swap in a new grammar, which is a much higher level specification. And what's more, the grammar could easily be augmented with specific features of interest in special cases. A particular aspect of tRNA for which grammars turn out to be superbly well suited is the presence of base pairing, which may be conserved across tRNA genes even when primary sequence isn't. So folded structure may seem like structure in a completely different sense than gene structure, but in fact, RNA base pairing is easily represented by what's called a self-embedding grammar. This one that you see here says that a non-terminal P rewrites as a P with complementary bases on either side of it, or it can rewrite to the empty string denoted by an epsilon and disappear. So the self-embedding rule can be applied recursively until such time as we invoke the epsilon rule, which terminates the derivation. There's obviously many such derivations, typically depicted as you see them here with double arrows. And the set of all such derivations derivable from a given grammar is defined to be the language generated by that grammar. So to show this derivation a little more graphically, we begin with the starting non-terminal P, and applying the rule once generates one base pair, which we connect with a red line to indicate their dependency. And doing so again repeatedly generates nested pairs of complementary bases until we invoke the empty string. And we end up with the dependencies nested one inside the other. In fact, the resulting parse tree actually displays a stem structure similar to the way it would be typically drawn. You can add details like loops, of course, but also mismatches, bulges, and so forth, all easily incorporated into grammar. I'm going to keep it simple here for demonstration purposes. Just think of these as idealized secondary structure. And we can easily add a rule that doubles the pair symbol, as you see here, and we apply that here in the third step of the derivation shown. And this allows branching, and thus the generation of arbitrary non-orthodox secondary structure. In fact, all possible folds in this idealized model. Now, more practical grammars for specific folded RNA elements can include primary sequence information as well in order to build pattern recognizers and so forth, using the same parsers for gene finding as I mentioned. In this case, ambiguity is also important since it captures the potential for alternative secondary structure. So parsing this particular sequence, GAUC, GAUC, can either generate a simple stem where we never invoke the doubled P rule, or a dumbbell structure where we double the P once at the very beginning. And note that the dependencies shown in red are no longer all nested within each other like Russian dolls, but we still call them nested in the sense that the red lines never have to cross one another. Yet another possibility is a cruciform structure where we invoke the double P rule twice. The grammar thus captures the structural ambiguity of RNA folding, but it also happens to be degenerate insofar as it might produce the same structure with more than one derivation, which is the case with this cruciform. But we can easily modify the grammar. Linguists call this lexicalizing a grammar, so that there's a one-to-one correspondence between derivations and unique structures. So this then allows for counting folds in this model system, and characterizing fold spaces in this way could be useful. For example, in designing sequences to achieve a specific fold with the least ambiguity, avoiding possibility of other folds with lesser or similar energy. And here I'm showing you just a glimpse of results from a prototype I did along these lines at the suggestion of Ard Lewis, which used this grammar to characterize all possible minimum energy folds of all sequences up to 10 mers, again, in this idealized system. Now, I need to make one more somewhat subtle point about ambiguity. We've seen that a context-free grammar can permit alternate folds, as in the case I'm showing you here, but if we happen to have a language that doesn't just permit but requires alternative folds, that turns out to be beyond the capabilities of a context-free grammar. And that's simply because the requirement to be able to assume either fold effectively creates crossing dependencies, as you see here, since some of the bases need to be reused, as it were. Well, alternative folding of RNA is indeed mandatory in some cases. For example, bacterial attenuators that control expression with a mechanism that depends on alternating hairpins. That is, a stretch of the nascent mRNA that's able to form a double hairpin is, under certain circumstances, also able to form a single hairpin, an alternative that modulates the continued transcription of the message. Riboswitches, another example, also alter their folds, typically based on interactions with small molecules. And that's the key, in fact. Switches in biology depend upon the existence of accessible alternative states, which are often established by alternative folding schemes, that is, through ambiguity. Just as ambiguity in language is generally resolved by context, switching systems are by design responsive to their context. We've seen that it's sometimes difficult to resolve ambiguity in language, something that Carroll freely exploits. However, the literary critic William Empson, who helped invent the technique of close reading in the 1920s, didn't see ambiguity as a bad thing. Rather, he felt it was all but essential, especially in poetry. Thus, while fields like mathematics and the law may try to drive out all ambiguity from language, literature has to embrace it to achieve meaning at multiple levels simultaneously and to establish various kinds of crossing dependencies. And it turns out that we don't even need ambiguity to produce crossing dependencies, because they're the signature characteristic of so-called non-orthodox secondary structures, such as RNA pseudoknots. These consist of two stem and loop structures that are interleaved so that half of each stem resides in the loop of the other. And such structures can't be described by context-free grammars, which can only have a single symbol on the left-hand side of rules. But relaxing this requirement leads to what are called the context-sensitive grammars, said to be beyond context-free. And I'm showing you an example here that produces pseudoknots, but I'm afraid you'll have to take my word for it because demonstrating a derivation would be pretty tedious. And all this brings us to the famous, dreaded Chomsky hierarchy, which is the fundamental infrastructure of formal language theory. I'm going to spare you the mathematical results, but what you need to know first is that as you ascend the hierarchy, computing with and about languages becomes more complex and less tractable. Second, language complexity in this sense is all about the nature of the dependencies that have to be captured in the structure of the language. You may be interested to know that human natural languages are thought to be mildly context sensitive, which is about where my own results and others place the language of macromolecules. This explains, for example, why pseudoknots broke standard RNA prediction programs as they crossed that threshold from context-free to context-sensitive. So with this, Chomsky essentially founded the mathematical field of formal language theory, which deals in sets of strings of symbols, all very abstract, although Chomsky's own compelling interest was in the human capacity for language. And I myself spent some time trying out mathematical formalizations of just the basic facts of molecular biology, which I'm not going to talk about here, other than to note that a lot of those facts can be very neatly encapsulated in a uniform format, including such things as Chargaff's rule, Kornberg's nearest neighbor analysis, and the capacity of DNA structure for self-replication, which is here demonstrated in a trivial one-line proof. But the most mathematically pleasing aspect of DNA would have to be the way that reverse complementarity allows for dyad symmetry in so-called biological palindromes. And more generally, symmetry as studied in group theory is, of course, of great importance in protein structure and its determination. Perhaps it was the mathematician in Carroll that inspired in him a near obsession with symmetry, which is found throughout his work in many forms and varieties. Just an example here, Tweedledum and Tweedledee are depicted as mirror images of each other. And when Alice first encounters them, they're described as standing side by side, each with an arm around the other's neck. And they also have a verbal tick, whereby one of them will make an assertion in if-then form, and the other will then add an else clause preceded by the word contrariwise. So it's as if Carroll, the logician, is looking for symmetry in syllogism. And one of Carroll's fellow dons at Oxford, William Spooner, is thought to have been the model for the white rabbit. But his real claim to fame was another verbal tick involving the symmetric exchange of words or parts of words in sentences or phrases. For instance, when he meant to say drink is the curse of the working class, he might switch words and utter work is the curse of the drinking class. This became something of a game for Oxford undergraduates who were actually responsible for many of the more elaborate Spoonerisms attributed to him. Well, we see the same phenomenon in proteins in the form of domain swapping. For instance, diphtheria toxin occurs as a homodimer, which can exchange an entire domain between subunits in symmetric fashion. The subunits are identical twins, and though they're not mirror images, one can almost imagine them as Tweedledum and Tweedledee, arms on each other's shoulders. So what about protein structure? Compared to RNA folding, it would seem to be considerably greater challenge for any grammar-based description, given the importance of 3D conformation and the much richer vocabulary of interactions. But a lot of the complexity can be abstracted away if we're addressing specific questions. And that's just what Ken Dill did when he developed models to try to account for the fast hydrophobic collapse in the early stages of folding. He first reduced the vocabulary of amino acids to just two types, hydrophobic and polar. And then by viewing folding as a self-avoiding walk on a lattice, maximizing the number of pairwise hydrophobic contacts, he was able to explore various algorithmic approaches to the search problem for insights into kinetics and stability and so forth. And this is really not so very different in spirit to the idealized RNA secondary structure grammars that I described above. And in fact, one of my own modest contributions to this work was to introduce Ken to the late Aravind Joshi, with whom I studied computational linguistics at Penn. And they published several papers together, including this work that conceived of folding as parsing, that is, modeling the process itself and not just the results. They used an efficient chart parser working on an HP model and improved on Monte Carlo methods while capturing in the chart itself known relationships between slow and fast-moving proteins and folding routes. Now, at the level of side chain interactions, the complexity of dependencies in proteins is obviously much greater than that of base pairing, but still there are intermediate abstractions that may be amenable to linguistic analysis. Just one example, secondary structural elements were schematized in Janet Thornton's lab in the form of TOPS cartoons, which had reduced representations of topologies. And these could be linearized for the purposes of database search. And in so doing, various spatial dependencies could be preserved as annotations in a manner very much in the linguistic mold. In fact, there's been a lot of activity in the grammar community around formalisms suited to capturing supersecondary structural motifs in biology, which is of interest to them because such structures are typically beyond context-free. Now, we've seen that natural language and RNA structures are just full of ambiguity, and this clearly applies to proteins as well. For example, the linker between the domains of calmodulin twists into a long alpha helix in the context of calcium, and then in the presence of a target protein untwists again so as to bind to it. And here again, we see how ambiguity, that is the potential to assume different conformations, affords switching. Function follows form, indeed, but form depends on context. The idea of parsing as folding is interesting in the light of what linguists call garden path sentences. Take a moment to read these three sentences that you see here. Many people have trouble with the last one, but in fact, it's perfectly valid if you back up and reparse it with the word old as a noun and the word man as a verb. So if you got stuck at the end of that sentence, then you know what it feels like to be trapped in a local energy minimum. That's pathological alternative folding, like that we see with the amyloid beta peptide in Alzheimer's and other misfolding disorders. And what's especially remarkable in this case is how perfectly good alpha helices can become beta sheets in the right context. This is truly protein folding led down the garden path. Domain arrangements can also be analyzed in the absence of explicit grammars at a notional level. Here's a simple concatenation of two domains, and it's linguistically simple. It's characteristic of the lowest level of the Chomsky hierarchy, which are the regular languages. However, insertion of one domain within another, as in an embedded grammar, is a hallmark of the next level, the context-free languages, which we've seen quite a few of. Finally, a translocation between two domains such that they end up interleaved, like pseudoknots in RNA, is beyond context-free, as we've seen, and also much less common. In fact, what we see here is nothing less than a recapitulation of the first three levels of the Chomsky hierarchy. The behavior of languages under various string operations is formally studied in part by analyzing their closure properties. The language class is said to be closed under an operation if the result of applying that operation remains in the same class. For instance, if a context-free language remains context-free after you've applied the string operation. So by interpreting various biological operations on sequences as their corresponding mathematical operations on strings, I could show that the context-free languages are closed under replication and what you might call everyday operations like scission and ligation. And on the other hand, they're not closed under what we could characterize as evolutionary operations, such as duplication, inversion, transposition, all of which would promote the language in the Chomsky hierarchy, that is, make it more linguistically complex. So you can think of evolution as a driving force for increasing the sophistication of biological languages. Then one interesting result is that context-free languages are closed under cyclic permutation, which is an operation we do indeed see in proteins in the sense that certain folds exist with their primary sequence termini at different positions in the structures. For example, here's a couple of lectins with their N and C termini highlighted to show how they're circular permutations of each other. And we should note, though, that the mechanism by which these are thought to arise involves tandem duplications in the genome. So while the end results might conserve linguistic complexity, the route to get there may not. So let's consider Carroll's work as it relates to evolution. We know that he was well aware of Darwin. He even corresponded with him. He went so far as to create a game that he first called Natural Selection that was played on a chessboard, which in turn was a major theme of Through the Looking Glass. And critics have found all sorts of Darwinian themes running through that book. Evolutionary biologists have returned the favor by adopting the term Red Queen effect to describe a principle that co-evolving species have to continue to adapt just to maintain their level of fitness, much less improve it. So in writing Jabberwocky, we know that Carroll was consciously imitating Old English or Anglo-Saxon. For most of us today, Old English does indeed look like a similar sort of nonsense as in this line, which even has letters we don't recognize. But that same line of text evolved over time, as you can see from these different editions of the source, which is, of course, the Bible. It was recognized in Carroll's time that languages evolved from common ancestors, certainly from the time William Jones put forth his Proto-Indo-European hypothesis in the late 18th century. And as you can see from the quote, Darwin himself found the parallels between linguistic and biological evolution compelling. In this, he was influenced in his youth by the writings of Sir John Herschel. So you can build phylogenetic trees for words with a good dictionary. That is one that includes etymologies. Here's an example. The 5 words at the bottom look somewhat similar, but in terms of meaning, they seem to have little in common, perhaps a vague sense of having to do with the straight and narrow. But indeed, they all trace back to a common ancestor in the Greek language, meaning reed or cane. There's a gradual change on each branch of the tree in both spelling and meaning. And practitioners of the field of historical linguistics have studied a variety of specific mechanisms of change for both, much as we studied evolutionary change in proteins. There's some noteworthy correspondences in how spelling and sequence relate to semantics and function, respectively. For one thing, while most words and proteins are fairly robust to minor changes over time, there are cases where just a little tweaking can produce significant functional shifts. And words like these that have different meanings but are easily confused are called paronyms. And we recognize these protein sequences as paralogs, assuming they arose by gene duplication. The word paronym is used differently in different subfields, but by one definition, paronyms must have a common etymological origin, as is the case here. So Carroll had great fun confusing words. When the mock journal describes his curriculum, which he presumably means to say that includes reading, writing, various arithmetic operations, what he actually says is rather hilariously distorted by mutations and accompanying functional shifts. The most obvious source of lexical ambiguity is simply the fact that words can have more than one meaning, such that when you look them up in the dictionary, they have numbered alternative definitions. The differences can be subtle. For example, if I say that 2 people differ, I might mean that they're unlike each other or that they actively disagree on some point. Because one implies the other, this sentence isn't very problematic, but there are certainly cases where such ambiguity can be confounding, as we'll see. In a similar way, proteins can be multifunctional or pleiotropic. Again, the differences may be subtle, but there's now a large catalog of what are called moonlighting proteins, whose multiple functions seem utterly unrelated. Several enzymes involved in intermediary metabolism, for example, take on additional roles, often in different cellular compartments that may have to do with signaling or transcriptional control or even cellular architecture. Carroll spends a lot of time on lexical semantics, and as you can see from this quote, on polysemy in particular. So as I said, words can be multifunctional, as in the case of the French word différer here. English derives two different words from this one, defer and differ, which, as we've seen, is itself polysemic. But note something interesting here. Each of the English words has inherited a different one of the two meanings of the French word. And this appears to happen in proteins as well, in what evolutionary biologists call subfunctionalization. Also interesting is the fact that there's another unrelated word, defer, in English, which is derived from a completely different source in French. And linguists call these homonyms. And the way you can distinguish this from polysemy is that homonyms have separate entries in the dictionary rather than being numbered variants. This is a kind of convergent evolution to a common spelling. And one also sees convergence from different origins to a common meaning, which is closer to the biological sense of convergent evolution. And of course, such words are called synonyms. And finally, as a special bonus, we see that it's possible even among these related words to find antonyms, words of opposite meaning. I have a particular fondness for autoantonyms, words that can mean their own opposite. In this sentence at the bottom, for example, the meaning of with depends on which century you're talking about. And if you want to find an equivalent in biology, you need look no further than reversible enzymes. So in this context, let's take a little closer look at the nonsense words now in Jabberwocky. And through the looking glass, Alice asks Humpty Dumpty for help in understanding the nonsense, and he provides a useful commentary on their origins. He says that brillig means 4 o'clock because that's when you begin broiling things for dinner. Well, as bioinformaticians, the first thing we want to do here is an alignment. And there are several likely possibilities involving indels and/or substitutions. And depending on the relative cost we put on these operations, we might favor one explanation over another as most likely. If, however, Humpty Dumpty was trying to come up with the most parsimonious explanation, he might have done better. Again, depending on the relative cost of indels and substitutions, there are actually several other words in the English language that provide cheaper sources of brillig. And it's remarkable how many of them refer to various means of cooking. Makes one wonder if Carroll maybe didn't choose brillig artfully to allow for these ambiguous alignments. And if you think that's far-fetched, consider how Carroll's mind worked when he invented his famous word puzzles. Perhaps his most well-known was what he called doublets, also known more often today as word ladders, in which you're challenged to transform one word into another, a letter at a time, through valid intermediate words in the minimum number of steps. Thus, getting from ape to man can be accomplished in this fashion. His choice of ape and man, by the way, suggests he had evolution in mind, though it perhaps betrays what was a common misunderstanding of Darwin's work, that humans descended from apes as they exist today rather than from a common ancestor. And we see this same misunderstanding expressed in another of Carroll's books in an even more egregious way, where a character imagines that a mouse evolves to an elephant. So just how well Carroll understood Darwinism is open to debate, but Darwin himself might have preferred a variant on doublet puzzles in which the goal would be to find the nearest common ancestor to some set of words. And the evolutionary biologist John Maynard Smith made good use of doublets and perhaps implicitly acknowledged a similarity between language and biology when he coined the notion of protein space and demonstrated how the word word could be transformed to the word gene in small steps via sensible words. He was trying to counter an argument that was being made at the time, this was 1970, that functional proteins were too sparsely distributed in a huge combinatorial sequence space to allow for transitions between them. That argument quickly became directly connected to the intelligent design movement, and it also bears some interesting similarities to Levinthal's paradox, raised at that same time in the realm of protein fold space. In fact, there are plenty of word pairs for which no solution path is possible, because words actually are pretty sparse, especially longer ones. Carroll considered this, but he never seems to have entertained the possibility of indels, which really would have made the problem far too easy to make a good puzzle. But he was open to other operations, for instance, allowing for the permutation of letters. And this allows us to get from lead to steel, for instance, with a permutation at the penultimate step. He also invented a number of other games along similar lines. And some of these bear an eerie resemblance to algorithms that are used in bioinformatics for sequence search and comparison. Yet another of these word puzzles was one he entitled Syzygies. And this entails aligning matching substrings between words, then repeating as necessary so as to get from, say, walrus to carpenter, trying to maximize overlap in some sense. And again, there are intriguing similarities with iterative alignment methods like SciBlast that are used to pull in successively more remote protein families. By the way, syzygy means an astronomical alignment of heavenly bodies, but it's also used to describe the alignment of chromosomes during meiosis. Well, if you still find the connection to bioinformatics remote, just consider the trouble Carroll took to optimize his alignments. He worked on a scoring scheme over a number of years, trying to define and combine parameters in such a way as to produce the most aesthetically pleasing, that is, in some sense, maximal alignments. Eventually came up with this rather elaborate quadratic expression at the bottom, which is pretty remarkable for a parlor game. And you have to keep in mind that this wasn't published in a mathematical journal, but in a popular magazine called The Lady that still exists, still promotes the puzzle. Now, the Needleman-Wunsch algorithm, which is the most basic sequence alignment method, uses dynamic programming to minimize edit distance. That is the total number of substitutions, insertions, and deletions required to get from one sequence to another. And it's specified mathematically by this classic recurrence relation. But the same recurrence can be specified with a context-sensitive grammar, shown here. Each rule corresponds to a different string operation, such as substitution, insertion, deletion, and each rule has a cost attached to it, shown in red. A derivation then entails the non-terminal S scanning from left to right, with the parser finding an optimal, that is least cost, sequence of operations such that one string is input and the other is output. As noted previously, using a chart parser is the moral equivalent of dynamic programming with similar theoretical efficiency. Now, simple edit distance proved to be biologically naive, and later algorithms implemented affine gaps with a large penalty for opening a gap and a smaller one for extending it. And the recurrence relation specifying this is more complex, but again can be readily specified with a grammar. Instead of implementing the algorithm from scratch, the same parser can be used, just switching out one grammar for another. This is similar to you and Bernie's early work with Dynamite, which used high-level specifications to generate dynamic programming code. In this case, the grammar is interpreted rather than compiled. Now, a yet more challenging recurrence relation was required to afford local alignment. Smith-Waterman. And again, this algorithm can be specified and implemented at a high level, but with just a little practice, I think you'd find much more readable and tractable to design. The great advantage of grammar-based alignment, though, is that it's easy to add other more biological features to the rules, again, without diving into code. So returning to Humpty Dumpty's commentary, he then addresses the origin of the word slithy, saying that it's a combination of lithe and slimy. Well, a geneticist immediately recognizes that this is a recombination. In fact, Slithy is a double recombinant with 2 crossover events, and it's a homologous recombination to boot, with the original 2 words sharing the substring Li. No doubt that homology helped inspire Humpty Dumpty's explanation. Ever since, what linguists call blended words have also been referred to as portmanteaus, as Carroll did, as Humpty Dumpty did. And Carroll himself invented a number of others, but the only one that's made it into the lexicon is chortle, which is a recombination of chuckle and snort. Portmanteaus have become quite common and tend to be coined at a steady rate, especially lately in reference to celebrity couples. Now, recombination might seem at first to be beyond the scope of grammars, but all that's required is the ability for rules to look at more than one string at once, what are called multiplex grammars. Here's a grammar where the 2 strings are referred to by subscripts and superscripts to the non-terminals. The S rule simply shifts the S to the right on both strings, but at any time can invoke the R rule, which swaps the 2 strings top to bottom as it scans, and which can at any time switch back to the S rule in turn. So here's a derivation where the input is motor on the top and hotel on the bottom. The S first scans the strings across 2 positions, leaving them unchanged, then exchanges itself for an R. And the R then finishes scanning with the strings inverted, accomplishing the recombination. We end up with both motel and hotor, the latter of which, of course, never caught on. And if we want to control recombination rates, we can parameterize the rules again, but this time with probabilities that govern with what frequency the rules are invoked. And this constitutes a stochastic grammar. And that's a tool that's been very useful in both natural language processing and RNA secondary structure analysis and prediction. Also, elaborating the grammar with additional rules can make the whole process much more biological. Now, we can use a multiplex grammar to scan double-stranded DNA, but we don't have to stop there. Grammars can be extended to deal in sets of strings, so that we could have a rule like this that acts on a base pair to duplicate it, separating it into 2 separate base pairs, identical to the first, here shown separated by a horizontal line. So that a derivation on a short double-stranded stretch would look like this. Note that an epsilon rule can be added so that the S falls off at the end, leaving a set of 2 identical double-stranded strings. We can use such a grammar to simulate the famous Messelson-Stahl experiment. You'll recall that they used heavy labeled bases and density centrifugation to test several hypotheses about where the new strand came from during DNA replication. And these alternative rules that you see here model 4 hypotheses with the labeled bases in red. And as you know, the answer turned out to be semi-conservative replication. And you may have noticed that the non-terminals in these last few grammars scan the input strings continuously from left to right, much like processive enzymes. And indeed, we can use non-terminals to model such enzymes with the grammar rules specifying their activities. Here's DNA polymerase with its multiple functions, each separately described by an alternative grammar rule, mapping to a different domain of the protein as well. Quantitative aspects of the enzyme activities can be captured with probabilities on rules again, and these can be fine-tuned, for instance, trading off fidelity and processivity. And moreover, the particular structure shown here is a mutant with no proofreading activity, which we can model by simply removing that grammar rule. Likewise, for other modifications, such as Klenow fragment, from which the primer removal domain has been lopped off proteolytically. Great flexibility. In fact, all manner of enzymes can be modeled in this way and thrown together in the same grammar with suitable sets of starting strings to allow for the simulation of cellular processes. For instance, to model replication more fully, we start with a double-stranded DNA in which the grammar can recognize an origin of replication and then recruit a helicase primase complex. And what you see here is a graphical output directly produced by the parsing system, shown in snapshots. Letting the parser run, you can see a replication bubble form, and polymerase is laying down both leading and lagging strands of new DNA, with Okazaki fragments being joined by ligases and ending finally with 2 double-stranded molecules. I've also tried adding centromeres, telomeres, multiple origins of replication to model chromosomes throughout the cell cycle. And similarly, models of transcription and translation, or in this case, both together, can start with double-stranded DNA and generate wave after wave of mRNA, and from that polypeptides, asynchronously for as long as you want to let it run. All this requires a lot of fiddling with rules and parameters to handle boundary conditions and get reasonable processivities, error rates, et cetera. And here's just a quick glimpse of the latter grammar as it's presented to the parser, just to give you a sense of what the grammar looks like. Clearly, there's a steep learning curve, but it's still relatively high level compared to what it does. Finally, returning to Humpty Dumpty's commentary on word origins, he explains that toves are a strange conglomeration of parts. The illustration by John Tenniel shows creatures with features of badgers, lizards, and corkscrews frolicking among the other strange beasts. And these toves and other characters, like the mock turtle and the griffin, are chimeras that represent somatic as opposed to lexical recombination. As depicted in Tenniel's illustrations, the striking thing about them is that they retain individual body parts unchanged from their presumed forebears, making them mosaics. And this flies in the face of the prevailing scientific theory of the time called blending inheritance, by which the entire organism would have been a uniform average, as it were. Even Darwin bought into this, though he later began to have doubts. And at the same time Carroll was writing this, Mendel was publishing his theory of particulate inheritance, which unfortunately Darwin never saw, but which can be considered a chimeric view of the assortment of traits. Having described lexical recombination with slithy and somatic recombination with toves, Carroll goes one crucial step further. His looking glass insects are given names by means of lexical recombination, such that rocking horse combines with horsefly to become rocking horsefly. However, the insects that are named in this way are simultaneously incarnated as real chimeric creatures, as if one were determining the other. In short, Carroll seems to be anticipating the central tenet of modern genetics, that genotype determines phenotype. Now, an old developmental biologist like me can't look at these mosaic insects without thinking of homeotic mutants in Drosophila, such as in Tinnipedia, which is illustrated here from the front with the legs growing out of the fly's head where the antenna should be. Hox genes that direct the morphogenesis of the fly are largely collinear in the genome with the actual body plan. And this theme is repeated and elaborated with Hox genes and other species, so that the lexical specification of the genome is literally mapping in a linear manner to the somatic form. There did happen to be a crossover event, then it would be reflected in the organism as well. In other words, we see all sorts of mechanisms in Jabberwocky that seem to reflect biological mechanisms as well. Other words are formed by various other manipulations of words and parts of words. And this is something that historical linguists catalog all the time in the evolution of languages. The most well-known of these mechanisms is compounding, which combines words or functional parts of words called morphemes, which you might want to think of as domains. Also common are clipped forms in which parts of words are discarded. And as you can see here, both taxi and cab are clipped forms of taxicab, which is ironic because taxicab is in turn a blend from the early French term for a taxi, taximetre cabriolet. And on top of that, the word taximetre is itself a compound. I think you have no trouble finding exactly analogous kinds of domain operations in protein evolution. For example, David Eisensberg lab recognized such phenomenon in what he called Rosetta Stone proteins. Certain functionally related protein domains may be fused in some organisms and separate in others, as seen in these enzyme activities for the first two steps in proline biosynthesis. And having built a database of such fusions, they found that they could use it to predict otherwise unknown functional relationships. It's interesting that an exactly analogous technique called collocation analysis had long been used by linguists to study lexical semantics. In fact, they even had to develop the same kinds of filters to get rid of noise from promiscuous domains. Using such inferred semantic relationships, linguists build densely connected word nets, which we'd call hairballs. So with that, I'd like to thank you for indulging me in trying to convey this linguistic view of macromolecules and in showing you various ways to have fun with grammars in the context of molecular biology. Beyond that, whether or not I've convinced you that Jabberwocky is about genetics, I hope I've gone some way toward demonstrating that Lewis Carroll, at the very least, had the skills and a mindset that would have made him well suited to a career in bioinformatics. Thank you.
Thank you very much, David. That was a tour de force. So we do have time for a few questions, perhaps from others who are not going to meet with David starting in at 1:15. Either unmute or type your question into the chat, please. So David, let me ask a question with the goal of connecting to your very diverse career. Were you able to use these concepts at GSK?
To be honest, no. I think you had a similar experience to me, Stephen. I think once you arrive in the belly of the beast, you kind of have to pay attention to the immediate projects that are facing you. I didn't stop working on it, but I can't say that it had immediate practical application. I think if I was headed in that direction when I was at Penn before I decided to make the jump, in that I think there, at the time I thought, and I still think that there are possibilities for using grammars to have a much richer semantic context to things like sequence search. Grammars couldn't compete with gene finders primarily because homology became so important with the filling out of the genome. But as long as you were dealing with just a vanilla gene architecture, you could use the same code for everything. But when you start to get into variants of gene architecture, as in, for example, immune-related genes, then I think there was the opportunity for a much richer contribution of grammar style of theme and variation. But as I say, I just didn't get to that and had to pay attention to business.
Yes, well, the balance sheet, particularly if you were when you're in a small company, is a hard master. Do you have any thoughts to share with us about the AI success of AlphaFold 2 back in December that got a lot of press? Have you given that any thought?
I haven't looked closely at it, but just at a surface level, I would say I'm actually surprised it took so long. AI has been advancing so fast in the last decade with advances in neural nets, which were fairly disreputable when I was in AI. The focus was much more on expert systems and rule-based systems. And the same thing has happened in language. in speech recognition and so forth. There was a traditional competition in AI between what they called the NEETs and the Scruffies, and the NEETs wanted everything to be rule-based and top-down, and the Scruffies said, take whatever data you have and throw it into the pot and find what patterns emerge by whatever means. And that was very much the neural net approach. And that has frankly triumphed. And you see it in all kinds of fields. And so I think that's the wave of the future.
Thank you. That's helpful. We certainly have been following what happened at Google DeepMind with great interest. I'm particularly interested in what they might be able to do if there was much more public domain data for protein small molecule complexes.
Oh, yes.
And being able to do a better job of docking of small molecules to proteins, and even more importantly, the scoring of the tightness of binding. And we did see in the drug design data resource activity, we did in their blind challenges, looking at docking and scoring, we did see some early hints that AI is much better than a rules potential based computed energy of interaction based approach. And that probably doesn't come as a surprise to you.
Yeah, and again, frankly, grammars are more in the neat camp. The one thing I would say though is that because you can add probabilities and make stochastic grammars, they have become much more versatile, and they have been used as extensions to hidden Markov models, for instance, in order to do things that are much more data driven.
Well, thank you, David. I want to acknowledge what a treat this was for us, and give you 5 minutes to decompress and get somebody to drink before we talk to the graduate students at 1:15.
Very good. Thank you.
Thank you again. That was a great talk and a fascinating exposition.